Hello everyone, welcome to my YouTube channel. Today we are going to discuss about DNA origami. So DNA origami, you might have heard. Origami is nothing but a five-folded kind of structure, which is actually basically named by after some of the group of scientists in Japan. So DNA origami is nothing but a technique in nanotechnology that involves in folding down DNA molecules into specific shapes. So what is nanotechnology? First we understand that. So nanotechnology in the sense any materials if their size is less than 100 nanometer, okay, if the size falls in zero point 0.1 to 100 nanometer, their dimensions, if any one of the dimensions, like it can be the diameter or it can be the length or it can be the width. So any of these three dimensions, any one of the dimension is coming in nanoscale, then we call it as a nanomaterials. Now these nanomaterials will have different properties from the bulk. That is why we are calling it as a separate technology only. That is why it is called it as a nanotechnology. Why it is called as nanotechnology? Why you are when we are already having a lot of technologies? Okay, because of this size. And why this size is very important? Because this size plays a very crucial role. If any materials falls less than 100 nanometer, they will tend to lose their bulk properties, but they become very excellent super properties. For example, a conductor less than 100 nanometer will become a superconductor. Understanding? So, if for example, if the hydrophobic structure, okay, if it is there, and if it is its size is less than 100 nanometer, it will give you super hydrophobicity. That means whatever, if the imagine one particular material giving one property, like for example electronic property, optical property as X, so in bulk, so if when it falls with the same material, if you convert it to the nanostructure, it will give you 10x to 100x of that particular property. That means conductivity will be increased. If it in terms of the optical this thing, optical property will be increased. Okay, like that. Okay, even hardness can be increased. You can simply take a simple example. You take 1 meter stick, you try to break it, you can break it very easily. Then reduce half of the meter. Then it will be a little bit difficult, but still you can break. Then reduce it to the 10 centimeters suddenly. So it will be very difficult to break it. Okay, that means you can break it, but you will have to apply extra force. That extra force why you are putting, because as the size is reducing, its hardness is increasing. Like in a similar way, nanotechnology, every property, consider it, be it is a magnetism, supermagnetism we will get, be it is a conductance, superconductivity will get, be it is a particular optical property, excellent optical properties you will get. For example, for optical properties, gold is there. Now what is the color of the gold? It is kind of yellow, right? You can don't say it as a gold, yellow color, right? So now if you convert into the gold, if you heat it for 1000 degrees Celsius, it will give you the nanoparticles. That means the bulk gold will be reduced to the nanoparticles. Now those nanostructures will have different optical property. That means if less than 100 nanometer, if it falls, the gold will start giving you the red color, purple color, less than 10 nanometer. Okay, so like that, understanding why this color changes, because when it falls less than 100 nanometer, the materials will there giving the excellent properties. The reason behind it, there are many reasons, mainly surface to value ratio. As you are reducing the size, what is happening? Surface becoming more. So you are getting the more active atoms on the surface. So you are having the different properties, understanding. So and then surface to value ratio over, and coming to the energy bands. In bulk, we are having the continuous energy band, whereas in nanomaterials the energy bands will be discrete in the structure, so that the conduction will be faster. So that was about nanotechnology. Let's start the topic DNA origami. DNA origami is nothing but it's one of the technique in the nanotechnology. Now you know what is nanotechnology. That involves folding the DNA molecules into specific shapes. Now this particular DNA origami, what is this DNA origami? It's nothing but a technique. Okay, what is this technique? All of you know DNA. DNA is a double stranded DNA. We all are made up of DNA, right? Because of the DNA we are here, right? So this DNA, a particular this single DNA double structure. We know that DNA has got double structure. So in that one single DNA molecule is taken, and you need to keep on rolling it. Like for a different shapes. It can be the circle, spherical, different kind of shapes you make it. Now why you have to make it? For many applications. It can be used to create nanostructures. It can also be used to create, to study the different molecular interactions. Also for the drug development systems or drug delivery system. Now this particular DNA is a natural molecule. If it is put in the inside our body, there is nothing wrong in this. It is very biocompatible, biodegradable, all the properties that it has got. Instead of making the liposome or bio capsules, or you can say the nanoparticles to use for a drug delivery, if you are using the DNA as a drug delivery, then it will be very helpful. No, that's why this DNA, single DNA is taken, it is rolled in different shapes and made it as a drug carrier. Okay, this is one of the application of DNA origami. This is the whole process is called as DNA origami. The process involves in using a long single strand DNA. That means you are taking a single stranded DNA, long single stranded DNA, which is called as scaffold. Now here you are calling it as this as a scaffold. Don't get confused with the other tissue engineering scaffold here. Okay. So you are taking a single stranded DNA, you are using it. That is called as scaffold. Now to guide the folding of short. Okay, now you are having the single stranded DNA. Now we need to fold it, right? So to fold it, we are using the complementary DNA strands just for as a reference, so that we know that DNA is a double helical structure. It is having the AGTC, AGTC will bind complementary to each other, right? So now if you are following this complementary, so now you have this particular as a DNA which you want to fold it. Now I am taking the reference, this thing, complementary DNA, which will help to fold this, but it is not in use of DNA origami. Understanding? Now the complementary DNA strands will come. Okay, they will call it as a staples. It will fix to it and it will try to, this can be manipulated by us, so by the humans, so that we can fold this like this, so that our DNA becomes spherical. Now this can be removed. Understood? Now your DNA origami will be removed. So DNA, you make it as a round shape or any other shape. Okay, make sure that it is in a kind of a circle or spherical structure. That is called as DNA origami. It is very simple. Okay, so here we are taking this DNA, we are calling it as a scaffold, and this DNA we are calling it as a staples. Okay, so enter desired shape, whatever the shapes you want, you can make it. Now the first DNA origami structures were developed in mid 2000. Okay, and since then the technique has been widely used in a variety of applications, including creation of nanostructures, the study of molecular interactions, and development of new drug discovery or new drug delivery systems. So as we discussed, creation of new nanostructures. Since what is the diameter of the DNA? It is 2.5 nanometer, right? So that is a natural nanomaterial DNA is. So this particular nanomaterial we can use to create the this particular DNA origami can be used to create the nanostructures. Next, study of molecular interactions. What are molecular interaction? When at the molecular, what is the molecular scale? It is between the atomic and nano, right? So what is nano scale? It is between the atomic and molecular structure. Now since you are coming with a nano, which is DNA origami, you can understand the molecular interactions very neatly. Why? Because it is having a size lesser than the molecule. So you can study the molecules at very closely, so that what exactly the interactions are happening in between the molecules, like sodium chloride. I'm giving as an example. A similar way it can be the biomolecules interactions, like protein-protein interaction, protein-nucleic acid interaction. Those all interactions can be understood properly by using DNA origami. Next, development of new drug delivery systems. Drug delivery system is what? Delivery systems is all of, to deliver something. Here, drug delivery in the sense, inside our body, like whatever drug we are taking as a tablet and all, it will be having 90% of sugar, only 10% of the drug. So by the time we eat it, it goes to the stomach and there is a lot of enzymes will be there, it will be degraded, water will be there, degraded, acidic condition will be degraded. By the time it reaches to the small intestine, it will remain only 10%. On that 10% only our body has to absorb. So understanding. So here what is happening? Drug is absorbing in the small intestine. Then only we are getting wherever it is going, and then we are getting it relief, be it is a fever or any other kind of conditions. But if you are using the drug delivery systems, we can avoid this loss of 90% of drug or any other glucose which is involved in it. We can load only 10% of the drug inside this, inside this particular DNA origami or drug delivery systems. Okay, and then you can directly inject inside our the nerves, or it can be taken orally. Oral drug delivery, transdermal drug delivery can be done, so that it will directly go to the specific point where the drug has to be delivered. For example, if it is a cancer tumor, it will go to the cancer tumor and deliver the drug. How it will go? Because these drug delivery systems will have their antibodies, okay, inside that, so that cancer cell will have antigen. Now this will antigen antibody interaction will be there. When you take the drug, it will go off finding the antigen. That is like, this is lock and key model. So it's like a couple, antigen and antibody. So now antibody I have in my drug. So its girlfriend is there in, now imagine this as a boyfriend. This girlfriend is there in a cancer. So it has to go and it will go and find its girlfriend and it will sit. So antigen antibody complex will form. Then the tumor will internalize this particular drug. So drug delivery system, and when it internalizes the internalized tumor, it releases the drug there, so that this particular cancer will be killed from inside, so that we can avoid chemotherapy, loss of hair, and we can also avoid the effects on in different parts of the body. All those things can be avoided by using this technique. So that's why in the development of new drug delivery systems, also DNA origami can be used. What applications? Creation of nanostructures. Second, study of molecular interactions. Third, development of new drugs delivery systems. So now the technological importance of the DNA origami. Some few other topics, the information what I just gave in introduction will be repeated here. Now the technological importance of the DNA origami lies in the potential to be used in wide range of applications. Now wide range of applications in the sense we are having the nanotechnology, that I explained you. Material science, you all of you know what is material science. It is normal mechanical engineering what you study, so that itself is material science, or whatever the science is involved in the material, that is called as material science. And biomedicine, so far drug delivery, drug discussion, whatever I study, that is about biomedicine. So nanotechnology, material science and biomedical application, it has got a lot of applications, that is DNA origami. Now some of the key ways in which the DNA origami can impact the technology is, the first thing is nano scale manufacturing. We can create the nano structures based on the whatever the size we want. If I want 10 nanometer, I can have. If I want 20 nanometer diameter, I can have. If I want 100 nanometer diameter, I can have. So that is why DNA origami can be used as a template for precise in the sense specific. I want this, so I will get this. So to precise assembly of the nanostructures, which have the applications in the various fields, nanomaterials, which are in electronics, photonics, material science and all, it has got applications. In your FET, field effective transistor, quantum dot FET is there, graphene FET is there. All these are nanostructure based FETs only. Next, drug delivery system. I have explained you what is drug delivery system. New approach for drug delivery, as it can be designed to carry therapeutic agents. Therapeutic agents are nothing but the drugs, okay, directly to the specific cells or tissues. This all technology has been success in preclinical trials in US, European countries, including the Germany, Sweden, Switzerland, okay, even in the Middle East like Dubai and Saudi, there are a lot of researchers are going on recently, and in Japan there is excellent work has been going on in Japan based on this particular drug delivery systems. They are using the drug delivery system so that we can avoid the loss of the drug and any adverse effects of the drug. So we can directly deliver the drug to the specific area where it is required, specifically in cancer related issues. Next, biosensors. Biosensors, all of you know what it is. So DNA origami can be used to develop the new biosensors that can detect specific biological molecules and also signals in the real time. So any signals, biosignals, if you want to detect, that can be used. The DNA origami can be attached to the normal sensors, so that will act as a biosensor. Now specific biomolecules, for example, if you want to detect a particular antigen, particular enzyme, particular protein, particular nucleotide, particular any kind of biomolecules, you can have this particular DNA origami on your biosensor, so that it can detect very easily since it has got biological property. Next, biomedical imaging. So it is very wonderful material can be used in the tool of biomedical imaging, so that it can it can be designed to target specific cells or tissues. To target specific cells and tissues, drug delivery system, how it works, the similar way it works here, so that we can target the particular cell like a cancer cells or cancer tissues or cancer tumor, and we can study that cancer tumor in the in diagnosis. So diagnosis in order to detect the cancer cells or to detect any diseases, and it also provides high resolution images, the real time high resolution images you will get in bioimaging. So next, gene therapy. Gene therapy DNA organ can be used to deliver the deliver vehicle for the gene therapy. That means it will act as how it is using. It has been used as a drug delivery system. Similarly, it can be used for a gene delivery system. So gene delivery is nothing but, so gene therapy is nothing but to imagine a particular person is not having from the birth only he can't see. That means he is blind. He means blind in the sense there is a particular gene which is coding for his vision protein is lost in his gene. That's why he is blind from birth. So now we are engineering his gene. We are replacing that defected gene with a new gene, so that he will get that protein, so that he will get the vision. That is called gene therapy. Understanding? Now to replace this particular gene, we need the delivery systems. For that delivery system, DNA origami can be used. Since it is a DNA, it can be used very easily. Next, biocatalyst. So it will involve in to develop new approaches for biocatalyst to perform specific chemical reactions. You know that enzymes catalysts are nothing but it can facilitate the reaction. Like that, as a biocatalyst or DNA origami can be used. Next, nano patterning. Nano patterning is nothing but again to create or to pattern or to fabricate where how exactly the nano patterning should be there. For example, microchips, nanochips for your electronic gadgets like supercomputers, quantum computers, and all you need. So for that, DNA origami can be used as a tool for the nano patterning. So it can be programmed to arrange and position nanoscale structures with precise control, precise and specific, specific whatever the size and shape you want, we can have. So those are about the important technological importance. Advantages. First one is programmability. That means we can have control over how exactly the folding should be done. With its precise and controlled folding of DNA molecule will be there into specific shapes or required space. What space we need, that shape can be we can get, so which can be programmed to fit the requirements of particular applications. Next, versatility. So DNA origami can be used to create a wide range of shapes. For example, 2D dimension shapes, 3-dimension shape. It can be used for many applications. Next, high precision, capable of creating nanoscale structures with high precision and accuracy. Right, useful for many applications in the field of nanotechnology. Next, functionality. So you can use this particular DNA origami structures. You can attach with some different structures. Functionalization using nothing but you are attaching something, so that then only it will become more specific function. So you can attach different molecules, additional molecules or materials, for example like proteins, nanoparticles and other materials, which can make them very useful for a variety of applications. Next, biocompatibility, that we have studied so far many times, it is repeated. So that the biocompatibility is in the sense when you are using something inside our body, we should check for biocompatibility, and it means that when you use it inside our body, our inside organ cells or anything should not get any effect from that material. So that is called, that's why it will be helpful for drug delivery applications and other bioimaging applications and other biomedical applications. That was about advantage. Limitations, complexity. Obviously, since the DNA structure, creating this particular DNA origami structure is highly complex, it is challenging, and it takes time, and also it requires special knowledge and even expertise. Okay, all these things are new. Expertise should be there. Next, cost. Since the producing and synthesis of DNA required a DNA origami, that can be very high, so making it as very expensive technique. Next, stability. So here we know that DNA is nothing but again which is natural molecule. Natural molecules are always fragile. That means they are very smooth, they are not so hard and all, right? So they are very fragile, so they can be degraded by the enzymes or any other factors which can limit their stability and shelf life, like temperature, pH, and etc. Next, scalability. Now that suddenly I want to make million copies of DNA origami, that cannot be done. So scalability is a challenge. So large quantities of complex DNA origami structures is difficult to form, and it is highly expensive, and also it requires a lot of time, money, and energy. So that was about limitations of DNA origami. So biocomputing we will discuss in the next class.